Hello, everybody. Welcome to IdeaStream 2017. I'm Kashif Khan. I'm a postdoc at MIT in the labs of Professor Buchwald and Professor Brad Penaludi. And I'm super excited to be sharing with you our research, which enables the development and design and development of smart drug delivery systems for cancer therapeutics. That's what we're working on. So one might ask, what does a smart drug delivery system entail? What are its components? Well, there are three components of it. I call it a smart design triangle. Element one is a smart recognition system that finds its target. Element number two is payload. Variability. We can find different, perhaps toxic agents that will essentially kill the target. And the third, and one of the important ones, sometimes understated, is a drug delivery mechanism, which holds everything together and then makes sure that the integrity of the whole system is not compromised. That's very important. There are, at a macro scale, these principles are already being employed in smart munitions to smart cars. In terms of the cancer therapy, ADCs, aka antibody drug conjugates, are basically the smart bombs at a molecular level to kill cancer. What are the components? As I mentioned, the navigation system that we recruit for an ADC is an antibody. This is nature's own targeting system to find a cancer tumor. Then we combine it with essentially a cytotoxic payload, which is too toxic to be used in any kind of therapy except that we attach it with a navigation system. So that's important. And the third part is the delivery mechanism, a linker that is competent enough to keep everything together inside the blood until the antibody finds its target, gets encapsulated inside the cell, and then the linker is designed such that it lets go of the drug, which then kills the cell. And the integrity of this whole system is very important. There are two ADCs in the market that are designed on these principles. One of them is Kadcyla that I list here. It targets HER2-positive breast cancer, which is basically 30% of the breast cancer type in women. So very important. Kadcyla, how it works, is essentially upon diagnosis of cancer, a patient is given an ADC, which then scans the whole body for the tumor. Then, once it finds the tumor, it attacks it altogether and then kills that tumor cell. And so this is far-reaching consequence. Turns out that in clinical studies of Kadcyla, 32% of drop in mortality rate was observed. That's significant. That's 32 people not dying out of 100. Very important. The problem, however, was the major side effects. Major side effects ranging from nausea all the way to huge toxic effects. So we think that these cytotoxic effects originated from the instability of the linkage that I show here. It's a traditional, typical linkage that people link an antibody with the drug. It's typically used. Both of the ADCs that I just mentioned involve this linkage. And it's unstable. It's known to be unstable. It's reversible, and so it comes off. And premature drug release, what would it mean? Not just the drug loss, but it would mean low quality of life for the patient. It would mean a lot of pain. So what we do at MIT is we have come up with a solution here. We have come up with a solution wherein we take our drugs, put them on antibodies through this blue linker, this aryl linker technology, and they are stable. Stability would then translate into broader therapeutic window, means minimal side effects, integrity of ADC not being compromised through dosage, so all those things. So And how do we access them? We access them through proprietary palladium reagents that are highly air stable and can be stored indefinitely, which is very important when you're in the lab and you have to make different analogs of something, very important. And you have to probably change different antigen. You have to target not the breast cancer, but some other sort of cancer. So you can recruit different antibodies, libraries of them, and then see where they are. We were interested in breast cancer, HER2 positive, and we wanted to develop a linker. Here's just a simple data point wherein we took our simplified, on the top, aryl linker with an MMAE, which is highly toxic drug, and compared it with the marketed linker. And here's the data. Here's the experiment. The experiment was simple, taking rat plasma in vitro and keeping it under physiological condition, 37 Celsius, and subjecting it with the two ADCs, our ADC with a very simplified linker versus a marketed linker ADC, and monitoring its integrity over 7 days. Data could not be clearer. As you can see, on the top is the blue line, where, as you can see, over 90, there's greater than 95% of the drug intact over 7 days. However, at the bottom, I show 60% of the drug has been released in the bloods. What does that mean? That's significant. That's important. And so that's the kind of stuff we wanted to show initially, that our linker does have some relevance to this kind of problem. Does changing the stability change the profile, the selectivity, discriminating between binding to tumor versus a healthy cell? Here's an example. On your left, you see the normal human kidney cells where we compare the control. And as you can see, only the highest dosage, which wouldn't be possible with our ADCs, will kill the healthy cells. So under normal circumstances, they do not. They're highly selective. But they are known to, they actually do kill the breast cancer cells. So that's the selectivity. And we can dial up that potency against breast cancer by adding up to 8 drugs, essentially. So that's where we stand. And currently, we are developing new linkers, new drug delivery systems. Based on that very simple aryl linker, the primitive form that I showed you, we're working on far more complex than that. And currently, we're thinking of xenograft studies going into animal, where we're collaborating with Dane Wittruph's lab at Koch Institute. And finally, the future, we are looking for essentially partners, and hopefully we can commercialize this technology and solve big problems in cancer. This is our team, 2 MIT faculty members, and a group of grad students and postdocs. With that, thank you so much for hearing us, and please stop by our poster, and we have other information to share with you guys. Thank you.